Okay, let's just picture the human body's defense system for a moment. Right at the heart of it, you've got these amazing Y-shaped things, antibodies, right, or immunoglobulins.
That's right, immunoglobulins.
And they kind of look like, well, a Y. Two heavy arms, two light ones.
Exactly. And those arms form the Fab regions. Think of that as the business end, the part that actually grabs onto invaders like viruses or bacteria.
Like a specific key for a specific lock.
Precisely. And then there's the other part, the FC region, the base of the Y. That's what signals the rest of the immune system. Like, hey, problem over here, deal with it.
It's such a clever design, really, how it recognizes things and then, triggers the response.
It absolutely is. And what's, well, quite remarkable is how the biopharma industry looked at that and thought, we can use this.
Right. They basically copied nature's homework.
Sort of. Over the last, 40 years or so, they've engineered these things called monoclonal antibodies, MAbs, based on that natural blueprint.
Monoclonal meaning they're all identical copies targeting one specific thing.
Exactly. And initially, the focus was really on the main type, the IgG antibodies. IgG1, IgG2, IgG4, those were the workhorses for the early drugs.
But it hasn't stopped there, has it? Feels like things have gotten way more complicated recently. It's not just that simple Y shape anymore.
Oh, not at all. The field is moving incredibly fast. We're way beyond the traditional format now.
So what are we seeing?
Well, you've got bispecific or even multispecific antibodies. Imagine an antibody with different hands, each grabbing a different target at the same time.
Okay, multitasking antibodies.
Kind of, yeah. Then there are ADCs, antibody-drug conjugates. Those are really clever. The antibody acts like a guided missile.
Delivering a payload.
Exactly.
Right.
Delivering a toxic drug payload right to the cancer cell, for instance, while minimizing damage elsewhere. Super targeted.
Wow. What else?
Then you have smaller bits, antibody fragments, things like Fabs, scFvs, even tiny ones called nanobodies. Their small size can be a big advantage, maybe getting into tissues better.
Right. Different tools for different jobs.
And finally, you've got these sort of mix and match versions, fragment FC or appended IG formats. It's like building with molecular Lego, combining different parts to get new functions. It's a really dynamic space.
It sounds incredibly innovative, but honestly, a bit overwhelming.
Yeah.
How does anyone keep track of all these different formats, what they do, which ones work? Sounds like a huge challenge.
It absolutely is. That's the crux of it. This explosion in complexity means we desperately need, better ways to organize all the information.
Right. You need databases.
Comprehensive databases, yeah. Ones that don't just list the antibodies, but tell you about their properties, how they work, what they're used for. It's crucial for R&D.
I know there are some databases already out there. I think I've heard of IMGT. And Drug Bank. There's ABDAB. Do they not cut it?
They're valuable resources, definitely.
Right.
But they often have, let's say, gaps. Maybe they lack some of the deep biological detail researchers need, or they don't integrate all the clinical trial info or the regulatory side or even the business intelligence aspects. It's often not the complete picture you need today.
Okay, so there's a need for something more.
Exactly. And that's where a platform like MAbFactory comes in.
MAbFactory. Okay, tell me about that. What's the story there?
It's a web platform from a company called MAbSilico. They specialize in computational antibody discovery, using computers to help find new antibodies.
And MAbFactory is their database.
It's their huge, comprehensive database. We're talking over 95,000 antibodies listed.
95,000.
Yep. Covering over 3,200 different biological targets. And crucially, it's not static. They update it every single week.
Wow. Where does all that data come from?
A lot of sources, but a big part is their analysis of patents. They've apparently dug through and curated data from nearly 3,500 patents.
Okay.
That covers something like 1,200 human targets where there's at least one antibody described in a patent that binds to it. Their goal is really to be the go-to place for navigating this whole complex antibody world.
That's a massive undertaking.
Yeah.
So for someone trying to get a handle on this field or maybe research a specific area, how does MAbFactory actually, help? What can you do with it?
Well, one of the main tools is called the Target Explorer. Let's say you're interested in a particular protein involved in a disease. You probably know it's UniProt ID, right?
Yeah.
That unique code.
Mm.
You just pop that into the Target Explorer, and bam, you get a whole dashboard of information about that target.
What kind of info?
You get the basics on the protein itself, quick links to that. But also, crucially, you see the competitive landscape, who else is working on this target. And, of course, a list of all the antibodies in their database known to bind to your target.
Okay, so a target dashboard page, what's typically on there? Break it down for me.
Right, so on that target's main page, you'll find things like all its known names, gene details, which species have it, where it lives in the cell, protein family,
Got,
functional bits called domains.
got it. The protein's ID card, basically.
Yeah, pretty much. Plus, what other molecules it interacts with, any 3D structures that scientists have figured out and put in the PDB, the protein data bank, and really useful, any antibody drugs already approved that target it.
Okay, that's super helpful context. And the competitive side.
That's there too. An overview of publications about it, clinical trials involving it, antibodies mentioned in patents against it, and the main disease areas people are targeting it for. It gives you that snapshot of who's doing what.
That sounds incredibly useful for just orienting yourself.
Yeah.
But what if you need to really drill down into the protein specifics?
Then you'd go into the target information and protein details section. That's where the real deep dive on the molecule happens.
Like what?
You get the full amino acid sequence, details on different versions or isoforms if they exist.
Isoforms, like slight variations.
Exactly, from things like alternative splicing
Yeah.
and PTM's post-translational modifications. That's stuff done to the protein after it's made, like adding sugars or phosphates, which can totally change how it works.
Right, those details matter.
Hugely. Plus, you see the defined domains, the 3D structure visualizations if available, even info on related proteins, homologs. Understanding all that is just fundamental to figuring out its biology and whether it's a good drug target.
Makes sense. And that competitive landscape section you mentioned, how valuable is that for, say, a company planning its strategy?
Oh, it's critical. It lays out who's active around your target. You see patented antibodies, published ones, what trials are running, the main therapeutic areas, which companies are in the game.
So you can see who your rivals are or maybe find collaborators.
Precisely. It's vital for assessing commercial potential, identifying partners, or just seeing if someone else has already patented something similar to your idea. Pure competitive intelligence.
Okay, and then the antibodies section, that's the list of actual antibodies that hit the target.
Yep. That lists all the antibodies MAbFactory knows about for that protein, and crucially, it gives you details for each one.
Right, the details.
Things like their sequences, how tightly they bind the affinity.
Affinity, right, like how sticky it is to the target.
Exactly. Also, where on the target they bind the epitope and any known functions, like does it block the target or activate it?
So you can actually compare different antibodies side by side.
That's the idea. You can see which ones have the properties you need, maybe high affinity or a specific epitope, and select the best candidates to look into further.
You also mentioned quantitative data, lots of numbers. How does that play in?
Yeah, that's a big plus. They've curated a lot of hard numbers. Over 27,000 affinity data points, that's actual measured binding strength, for over 16,000 unique antibody variable domains.
Wow, that's specific.
Very. Plus over 10,000 epitope data points mapping where they bind. Cross-specificity data for 5,000 domains does it bind to similar proteins, maybe in other species, and over 18,000 functional measurements like EC50 or IC50 values.
Which tell you how potent the antibody is.
Right, how much you need for an effect. Or things like TIM, which relates to stability. Having all this quantitative data lets you do much more rigorous analysis. It really enables data-driven decisions, not just guesswork.
Okay, I'm starting to see the power here. Could we maybe walk through an example? You mentioned VEGF-A earlier, vascular endothelial growth factor A. Can we explore that using MAbFactory's tools?
Absolutely. VEGF-A is a perfect example. It's a polypeptide, 395 amino acids long. Its main job is driving the formation of new blood vessels.
Angiogenesis.
Angiogenesis, exactly, and also vasculogenesis, the development of the whole vascular system. You might also see it called VPF, vascular permeability factor.
Why is that?
Because it also controls how leaky blood vessels are, how easily stuff passes through their walls. So yeah, it's a master switch for blood vessel growth and function.
And does it have those complexities you mentioned before, like isoforms and modifications?
It sure does. VEGF-A has multiple isoforms because of alternative splicing. The gene can be read in slightly different ways. The longest form is the most common.
Okay.
And it definitely gets modified. Disulfide bonds form, which help hold its 3D shape together, and it gets glycosylated sugars get attached. These mods are important for its function and how it interacts with things.
Got it. And structurally, what are the key parts of VEGF-A?
Well, there's a signal peptide that tells the cell where to send it. Then the really important bit is the PDGF-VEGF domain. That's what binds to receptors on other cells to kick off the blood vessel growth signals.
The business end again.
Pretty much.
Yeah.
It also has a heparin binding domain, which lets it interact with molecules in the environment that help regulate the whole process. Understanding VGFA is critical because it's involved in normal things like wound healing, but also in diseases like cancer.
Right. Tumors need blood vessels to grow, so they often hijack VGFA.
Exactly. That's why it's such a major drug target.
So mainly involved in blood vessels, growth, permeability. Where do you find it usually? And what's its family?
It's found mainly in the cytoplasm and nucleus of cells. It belongs to the PDGF, VEGF growth factor family, a bunch of related signaling proteins. Key domains are that PDGF, VEGF1, and the heparin binding one.
And what does it interact with? Receptors?
Yes, it signals through receptors like FLT1, KTR, and NRP1 on cell surfaces. And we actually know quite a lot about its structure. There are 53 high-res structures in the PDB.
Okay. And are there actual drugs targeting it now? Antibody drugs?
Oh, yes. Bevacizumab, brand name Avastin, is a big one. It's an antibody that specifically binds VGFA and stops it from working, used mainly in cancer.
Given its importance, I bet there's a ton of research. What does MAbFactory show for VGFA activity?
The numbers are pretty telling. Over 11,500 scientific papers on VGFA, over 4,300 clinical trials involving it.
Oh.
And MAbFactory's patent analysis has pulled out 944 different antibodies targeting VGFA mentioned in patents. It's a very, very hot target.
Clearly. And the specific anti-VGFA antibodies in MAbFactory, how many sequences?
They list 997 sequences for antibodies against VGFA in various formats. And for these, you get all that extracted info we talked about. Plus, you can filter the list, say, show me only antibodies binding a specific epitope or with an affinity above a certain level, or those with an INN, an official drug name.
Okay. You mentioned one specific antibody earlier, 97799. What's the story of that one?
Right. Antibody 97799, it's an IgG type. It came from a patent filed back in 2011 by a couple of entities. 529-16213-33681-0CB USA LLC, and the Development Center for Biotechnology.
Okay, and what's interesting about it?
What's compelling is the preclinical data mentioned in the patent. It showed good results in reducing tumor growth in an in vivo xenograft model, basically, human tumors grown in mice. That suggests real therapeutic potential.
And MAbFactory provides the molecular details for it.
Yes. Sequence analysis shows it's humanized engineered to look more human, less likely to cause an immune reaction.
Less immunogenicity.
Right. And crucially, MAbFactory provides the specific amino acid sequences for its heavy and light chain CDRs.
The CDRs are those loops that actually touch the target.
Exactly. The complementarity determining regions. They define the binding specificity. The database also lists its binding affinity, how tightly it sticks to VGFA. It's got a KD of 2.910 molar, measured by Biocore, which is pretty strong binding.
That's very specific data.
And they even identify the specific epitope, the exact landing spot on VGFA that antibody 97799 recognizes. So yeah, a lot of detail on just that one antibody.
Okay, so that's the target explorer, starting from the target protein. What if you already know the antibody you're interested in? You want to find everything about it.
For that, you'd use the Antibody Explorer feature. It does what it says on the tin, lets you search for a specific antibody, maybe by its name or even its sequence, if you have it.
And it pulls up a profile for that antibody.
Exactly. A comprehensive profile with all the info MAbFactory's gathered on it.
What's typically in that profile? Let's break it down.
Okay, you'll get general stuff. Alternative names, what target it hits, its origin, human, mouse, humanized, etc. Then, deep sequence info. Full, heavy, and light chain sequences with the CDRs annotated.
Annotated meaning clearly marked.
Yes. Plus, germline information, which original V and J gene segments from the immune system it's most similar to, with species and scores.
Okay, tracing its lineage almost.
Kind of. Then, binding properties, affinity data, KD values, epitope mapping details if known, functional effects, does it block, activate, neutralize, cross-reactivity, does it bind the target in other animals?
That's important for preclinical testing.
Very. And patent information. Patent numbers, who filed them. And finally, source details, maybe the cell line used to make it, things like that. It aims to be exhaustive.
That is incredibly detailed.
Yeah.
Could we use another example, maybe an antibody drug people might know? Daratumumab, perhaps?
Daratumumab, sure, that's a well-known one. It's a human monoclonal antibody, an IgG1. Used mainly in cancer, target CD38.
Okay, so if I search for daratumumab in the Antibody Explorer.
You'd pull up its profile, you'll see it's human IgG1, target CD38, all the basic info.
On the more granular stuff,
Yeah.
sequences,
Cool.
binding.
Yep. Full, heavy, and light chain sequences with the framework regions and those crucial CDRs highlighted. You'll also see the closest human germline V and J genes identified for both chains. And for daratumumab, MAbFactory has extracted a ton of data from patents. They mention analyzing 16 patents and applications related to it.
Wow. What kind of data came from those patents?
Really useful stuff. Cross-reactivity, it binds CD38 on cynomolgus monkey cells, which is useful for testing, but not mouse or rhesus monkey CD38.
Good to know.
Details on the specific epitopes it binds on CD38. Measured binding affinities, KD values for both human and cynomolgus CD38, and even functional data like EC50 and IC50 values from various assays, showing how potent it is against different cell types or in different functional tests. It's a really rich picture of that specific antibody.
It's clear this is a powerful database. Stepping back a bit, how does having all this information actually help in the day-to-day work of discovering and developing new antibody drugs?
Well, the platform, the software, and the database together, they're really designed to support a whole range of activities across the R&D pipeline. The goal is to speed things up and allow for better, more data-driven decisions.
Can you give some concrete examples of applications?
Sure. Take target validation right at the beginning. The Target Explorer is perfect for this. You get all that info on the target protein sequence, structure, function, expression, plus the competitive landscape. It helps you prioritize, right? Decide if a target is really worth pursuing based on the biology and who else is working on it.
Okay, so it helps you pick your battles wisely.
Exactly. Then, once you have potential antibodies, you need antibody characterization. MAbFactory provides the sequences, affinities, epitopes, functional data. You can compare candidates, select the best ones, and crucially, start understanding those sequence structure-function relationships.
And that epitope mapping data you mentioned.
Yeah, knowing precisely where an antibody binds can be critical for understanding how it works, why it has the effects it does.
Makes sense. What about the intellectual property side?
That's where patent analysis comes in. Seeing the patents and applicants related to antibodies and targets is essential for competitive intelligence, figuring out freedom to operate, identifying potential IP hurdles early on,
Right, avoiding stepping on toes.
or finding space to innovate. And if you're looking for new antibodies, the Find Binders tool helps with antibody discovery. It gives you a starting list of known binders for your target, which could be leads themselves or starting points for improvement.
Improvement through antibody engineering.
Precisely. Information on existing antibodies, their sequences, affinities, epitopes can directly guide how you design new ones. Maybe you want tighter binding, better stability, a specific functional effect. The existing data gives you clues.
And all this feeds into the bigger picture of drug development.
Absolutely. From target validation right through to clinical trial design. Knowing the competitive landscape, what trials are running, the main therapeutic areas, it all helps shape development strategy and assess the potential of your candidate.
It also sounds like this huge data set could fuel more advanced computational work, maybe AI.
You're spot on. That's algorithm development. This wealth of structured data sequences, affinities, functions, structures is perfect training material for machine learning models.
So AI could learn patterns.
Exactly. Learn the complex rules linking sequence to structure to function. This could lead to predictive models that help design completely new antibodies with desired properties, potentially speeding up discovery significantly.
It really is impressive how much is pulled together here. Seems like the integration is key.
That really is the core strength. MAbFactory serves as this incredibly comprehensive but also dynamic resource for anyone in the antibody therapy space.
Dynamic because it's updated weekly.
Right. It provides us deep insights, molecular details, target properties, the competitive environment. With over 95,000 antibodies and 3,200 targets covered, it's just a hugely valuable tool in modern medicine where these therapies are becoming so central.
So if you had to summarize the biggest advantages MAbFactory offers.
I'd say number one, the comprehensive data coverage on both antibodies and target sequences, binding, function, epitopes, all of it. Number two, the user-friendly interface with those powerful search options. Three, the integrated competitive intelligence tracking patents, publications, trials. And four, that wealth of quantitative data enabling really rigorous analysis and data-driven decisions.
And it's not a static snapshot. It keeps growing.
Absolutely. That potential for expansion and the commitment to regular updates is vital. The field moves so fast, the database has to keep up to stay relevant. As new antibodies emerge and move through development, the database grows with them.
So wrapping up, MAbFactory really seems designed to cut through the complexity, providing clarity.
That's the goal, to enhance understanding by bringing together comprehensive, detailed, dynamic data. Ultimately, that supports better decision-making and helps advance the whole field of therapeutic antibody R&D faster.
It really makes you think, doesn't it? Having all that data integrated and accessible. What kind of future breakthroughs could that unlock? What new treatments might become possible for all sorts of
This
diseases?
is an exciting prospect.
Definitely. If you, the listener, want to check out MAbFactory, head over to their website, www.mabsilico.com. And there's actually a public demo version you can play with. We'll put the link in the
Yeah.
show notes. It's app public demo MAbFactory 97288.azurewebsites.net.
That's great, a hands-on look.
Yeah. And for those interested in antibody databases more generally, the Antibody Society also has a relevant publication about their own database efforts on Biorex. We'll link to that too. This has been a really fascinating deep dive. Thanks for walking us through it.
My pleasure. It's a powerful tool in a really important field.